name is Haley Fitzpatrick, and this is my presentation over my protein research paper. The title of my document is Porin, a transporter protein. So first I started with an overview of proteins in general, about their functions, the structure. And protein, of course, is a crucial molecule. You can't live without it. They do a bulk of the work in the cell. They help with the function, structure, and a whole lot of different things. And the structure, there's 20 different amino acids, and these are linked by peptide bonds. They form long chains, and there's no breaking in between them. And the one thing that varies between them is their side chain. So this side chain, it can be nonpolar, it can be polar, it can be electrically charged, and this is the difference between the different amino acids. So, of course, 20 amino acids, imagine the amount of different proteins there can be from that. For all the different side chains and everything, those create tons of different proteins. So the diversity in protein structure and makeup, that creates many different functions. And just a couple of examples of some functions include antibodies, enzymes, messengers, structure, transport, gene regulators, and storage. And proteins have an exclusive complex structure that allows them to act upon their specific duty to keep the cell in operation. So as you will see throughout this presentation, function and structure coincide together and they basically work together. So like the structure is important towards the function. So now let's move on to the 4 levels of protein structure. So there's primary, secondary, tertiary, and quaternary. And of course, like I just said, the shape of a protein structure coincides with its function. And more specifically, the 3D structure, the tertiary, is probably the most important. It's the one that creates the function overall. So first, primary structure. This is the amino acids linked together. There's no branching at all between them, and they're linked by peptide bonds creating a peptide chain, polypeptide chain, I'm sorry. And then the secondary protein structure, these are beta-pleated sheets. There's alpha helixes, which create like a spiral. And these are brought together by hydrogen bonding. And next we have tertiary structure, which I don't know if you can really see too well, but this one is like the jumbled mess of it all. So it's in 3D, it's different winding, looping, there's many different beta-pleated sheets, there's many different alpha helixes, all combined, and it's only using one chain. So it's one chain, and they're reinforced by the side chain. That's how they hold together, and that's how they get their structure, mainly from the side chain. And then finally, quaternary. This one it's whenever there's more than one polypeptide chain used. So there's more than one chain, it's creating one protein itself, but two or more chains are used. So next, my specific protein I got chosen to do is Porin, and it's a transporter protein. So over here I added a picture of a transporter protein process. So Porin, it makes a channel out of beta-pleated sheets, and it's kind of cool. It's different than most. It has hydrophilic insides, and so only hydrophilic is very, very specific. Only specific molecules can go through the channel and make its way through by diffusion. And this is an outer membrane protein found in mainly gram-negative bacteria. It can also be found in chloroplasts, mitochondria, and myobacteria as well, but mainly it's found in gram-negative bacteria. And its cellular function is to regulate the passage of molecules through the outer membrane. Of course, it's very specific. And the function as an organism is to transport nutrients. And there are 2 main contributors to the function and specificity of an individual porin, which distinguishes which nutrients it will uptake. So, of course, reiterating that specificness of it is from the differing amino acid makeup and the pore sizes of the channel. So, of course, if a pore channel is really tiny, it cannot take in huge molecules. It can only take in small hydrophilic ones. And also, as a side note, Porin, I found throughout my research, is that it has a central role in pathogenicity and protecting bacterial cell by resisting and accepting certain antibiotics. So I thought that was an interesting fact as well. And now, this is Hiroshi Nakado. He is known as the porinologist, and he founded Porin in around the 1980s. And he had tons of different research he published different documents, he went through different experiments. An important one he did that I found was he determined the rates of diffusion through OMPF, OMPC, and PHOE porins, and these are main porins found in the gram-negative bacteria. And this research transformed the understanding of how antibiotics respond or resist, and as well as how the transport pathways worked within bacterial cells. So, great important discovery he had. And this there's a recent spark in finding more knowledge and gaining interest in porin because it plays a significant role in bacterial pathogenesis, and the recent research in spike has been because of this. And it could actually help with the development of future therapeutics. So this is important, and porin, of course, can help with that. And now to its structure. This is a ribbon model of porin, and it's filled with beta strands. It's only composed of beta strands, no alpha helixes at all. And I don't know if you can really see that well, but it's anti-parallel in structure, so it's going to be having different, like one's going this way, the other one's going that way. And that its secondary structure, the beta-plated sheets. And as you can see, it kind of makes a channel, so it's around a channel. It's a cylindrical channel, and it's known as the beta barrel. The tertiary structure consists of transmembrane barrel composed of the beta strands, and they're linked by different sized loops called short periplasmic loops and longer extracellular loops. So you can see the little loops around, and then of course the beta-pleated sheets. That creates the whole 3D complex structure of it. And the different colors, the red and green, it resembles the polar and non-polar disposition of the amino acids throughout. So they're alternating throughout. There's not just one type of amino acid. There's polar and there's non-polar. And here's a different picture of it. So this mainly highlights the inside core of it, which you can see the green is only shown there, which is the polar amino acids. That represents the polar ones. And you can see that there's no nonpolar at all. They're mainly around the outside. And that is due to a loop that's blocking those nonpolar ones from getting in there. So this right here creates that hydrophilic channel that allows the hydrophilic molecules to move through. So of course the aqueous channel is created by that. And porin, it has subunits, and it's known as a trimer. And a subunit is a single protein molecule that agglomerates to form a protein complex. And here it takes 3 identical porin proteins and creates one protein complex. And so that's known as a trimer whenever it's 3 identical ones composed into 3. And here it kind of shows the upper, you can see the channel between them, and they form like one whole thing. And then this shows the more of the ribbon model with the beta-pleated sheets highlighted as well. And of course, the different colors represent one porin from each other. And now to the DNA sequencing for porin. So I found one research document I read. It went over, it specifically talked about the OMP32 porin, and its structural gene encodes a precursor consisting of 351 amino acid residues with the first 19 amino acids constituting a signal sequence. And throughout I just put this one, but some of them are really similar to that with the signal sequence around that amino acid size, and of course amino acid residues. There's a huge variety, but mainly there was a lot of them as well. And these similarities and differences are of course due to different functions, different areas of the body they're in. Of course, if it's in a chloroplast or if it's in a gram-negative bacteria, they're going to be different structurally and have a different DNA sequence. And of course, going back to evolution, that's how they change as well. And I also found that there are 8 regions of homology, which are there's patterns between the primary structure of porins and their DNA sequences. So I do have a picture of that on the next slide. And as you can see here, there's the black little lines are the homologies between the different, there's like 1, 2, 3, 4, like 8 or so different porins here, and you can see the similarities between them. And the black bars indicate the homology, and the dotted bars are alternative homologies, so those are the differences. And of course the hatched bars show the signal sequences. So many similarities between porin, and that helps with the DNA and how they're similar. And there was not a ton of info on genetic diseases linked to porin, but I did find one. There's an anti-OMPC porin, and it can be used to help diagnose inflammatory bowel disease and as well distinguish between ulcerative colitis and Crohn's disease. And I found like probably but distinguishing between ulcerative colitis and Crohn's disease is probably the most important one I found. I found a lot of documents about that. And of course, the anti-OMPC is an important biomarker. And high anti-OMPC levels, of course, are seen in families with a history of ulcerative colitis and Crohn's disease. So you don't want a high-level anti-OMPC. And in conclusion, of course, proteins are important components to a cell's ability to do work. They're extremely important. You can't live without them. And porin and other proteins are profoundly complex polymeric macromolecules essential for a cell's ability to function. So porin's important, and of course there's many other proteins that are important in this as well. I just highlighted porin throughout this video. And I want to end on a quote that was in our book and in our lecture. I just think it overall concludes proteins in general. It says, from a chemical point of view, proteins are by far the most structurally complex and functionally sophisticated molecules known. And that's what honestly, that's like a summary of proteins right there. They're so complex and everything, and they have so many different functions. They help the body in so many ways. Well, anyway, thank you for watching. Don't end it yet, just yet. I want to show my literature cited. So here are a couple of and this is my literature cited here. Okay, thank you for watching.